We are talking about the cell is a very important topic for both grade 10 CBC and 8 for 4 system. So the meaning of the cell also is a common topic in examination. So the meaning of the cell is just a basic unit of structure and function of organism. So the basic unit of the organism is known as cell, meaning that that's the structure, the basic unit. That's where a human being starts. For example, when you are young, you are very small, you are very short, but now you are tall and you are big, meaning that your cell has been expanded or has been actively divided, and then you are where you are now. So meaning that the cell is the basic unit of a structure and the function in an organism. So the cell, these are when we are talking about organisms now, we have two type of organism: the unicellular and the multicellular. Unicellular organism are organism that has only one cell. So if organism has only one cell, are known as unicellular. While when organism has many cells, they are known as multicellular. So unicellular organism, they comprise of amoeba, paramecium, and plasmodium. These are unicellular organisms. Multicellular organisms are made up of many cells, the like of the moles, grasshopper, elephant, man, a human being like us, mouse, all of that they are multicellular organism. So a cell, it's a very small structure that cannot be seen by naked eyes within our normal eyes, meaning that they require microscope for them to be observed. For them to be observed, they require a microscope. So function of the microscope, it has only two functions. Number one is magnification, and number two is resolution. Sometimes in exam situation, you will be asked to define what is magnification and what is resolution. Magnification is just making very small organism to appear bigger so that they can be seen. You just make a certain organism to appear a very large, and for that you can see it and study very well. But resolution is just to the ability to distinguish two structures that are very close together as distinct entities. That's known as resolution. Meaning that these two things, they are very close. They are very close. They are very close in such a way that they have almost similar properties. They have almost similar characteristics, but that ability to differentiate that two structures are known as resolution. It's known as resolution. So type of microscope. In microscope, you have two microscope. Light microscope, mostly that is available in school laboratory, and we have electron microscope. So light microscope uses light. That's why it has mirror on it. It has mirror on it, so it has light. So the light will reflect here, then it will enter on the diaphragm to the stage, and then you will see it from here. So this is known as light microscope. Electron microscope, it uses electric now. It uses electric beam or electric rays. So when you're talking about part of the microscope, they are very common in exam, because sometimes you'll be asked some part of the microscope. It might be drawn, and then labeling might be asked in exam, or it may be asked in exam station. You might be asked to answer some questions based on their functions, or you might be given their function, and the examiner will tell you to write the part itself. So let's look in details about microscope. The upper part where you will bring your eye is known as the eyepiece lens. This is the lens. So in microscope we have only two lens. That is the eyepiece lens where you will place in your eyes, and objective lens where it's just above the clip. We have that two lens. Also, we have the body tube. We have the body tube. So we have the adjustment knob are two. Number one is coarse, which is the bigger one, adjustment knob. Number two is the fine, which is the smaller one. And another thing that you have is the arm, where we hold it. We hold it. That's the upper part. We have the eyepiece, we have the coarse adjustment knob, we have the fine adjustment knob, we have the body tube, we have the revolving nose piece, we have the objective lens. So that is the only thing that we have on the upper part. The lower part, we have the stand, that's the base. We have the mirror that reflecting for light. That is for reflecting for light. Or another thing, we have the diaphragm where the light passes. So we have the condenser. So diaphragm it hold the condenser. And we have the condenser. Then we have the stage. Then we have the clip. We have clip. So this is a part of the microscope. They are very crucial for you to know that. Especially some the parts that it's very important for you to know, and it might ask in exam situation. They are very common in exam. It is eyepiece, coarse adjustment, fine adjustment, the body tube, revolving nose piece, objective lens, the stage and condenser, very common, diaphragm and mirror, very common. Sometimes you might be asked their function too. So use of different part of the microscope. Number one is the limb, that is or the arm. What's the function of the arm? Is to support the body tube and stage. So its work is to just support the body tube and the stage. So it supports the body tube. This is the above one, and the stage. This is the stage. It supports that one. Number two is the base or stand. It provides stable support because without this base, the microscope cannot stand on its own. So meaning that the microscope requires a base or stand for it to for stable support or for it to stand alone. Number three is body tube. Body tube it holds two things. It holds the eyepiece and revolving nose piece. Look at it. Look at it. This is the body tube. So this is a body tube. So this is a body tube. In this body tube, look at it. It is the middle part of the eyepiece and revolving nose piece, meaning that it holds the eyepiece lens and revolving nose piece. Revolving nose piece. Another thing is the eyepiece lens. Eyepiece lens it contains lens for magnification. Simple as that. Cause adjustment knob is to just bring the image into rough focus by raising or lowering the body tube through long distance. So meaning that we have two adjustment knobs. Number one is the coarse adjustment knob. Number two is fine adjustment knob. Their function, the difference is only one. Number one, the coarse adjustment knob it brings the image into rough focus, while the fine adjustment knob bring the image into sharp focus. That's the only difference. It brings the image to sharp focus. The fine adjustment knob, the coarse adjustment knob, it brings image into sharp focus. And I think we have the diaphragm. Diaphragm, just when the light from the sun, the natural light from the sun hit the mirror, the light move through the diaphragm, meaning that diaphragm to condenser, meaning that diaphragm it acts as a mediator, or in other words, it controls amount of light passing through to the condenser or through condenser. So it just regulates amount of the light passing through the condenser to eliminate displacement. Objective lens also they have lens for magnification. Simple as that. Mirror reflects light through the condenser onto the stage. So that's the work of the mirror. It's just reflection. Revolving nose piece is look at it. Revolving nose piece is here. Revolving meaning that it's something that can revolve or rotate. So it holds the objective lens. It holds the objective lens so that uh, the viewer the viewer can enable to change from one objective lens to another, because this objective lens, it has different magnification, meaning that this can have 2x, this can have 3x. Look, it has different magnification. The same thing as your phone. When you enter your camera and then you try to zoom, you can magnify it by it give you different magnification. For example, it can give you 2x, 20x, 30x. That's the magnification. The same thing applies to the microscope. Objective lens has different magnification. Another thing we have is the condenser. It concentrates light onto the stage. Condenser, very common question. Condenser concentrates light on the stage. Stage is a flat platform where the specimen on the slide is placed. So stage is just flat platform where specimen on the slide is placed. Also, we have the clip. Clip is just to hold the slide in position, and that's all about the function of the microscope. Handling of the and the care of the light microscope. So when you're handling a microscope, you must handle with care. Number one, always use both hand when carrying a microscope. Number two, never place microscope too close to the edge, meaning that if the table is here, don't place the microscope too close to the edge in such a way that it can fall. Number three is not touch the mirror and lenses with your finger. Don't touch the finger and the lenses on your finger, because it will make it dirty. It will make it dirty, and then you will not see it very well. Dirty lenses should be cleaned using special soft lens tissue paper or tissue paper moistened with ethanol to avoid scratches. Number five, that part of the microscope may be cleaned using a soft cloth or tissue paper. Do not wet any part of the microscope, because it will lead to rusting. It will lead to rusting. Number seven, make sure that the low power lens clicks in position in line with the eyepiece before and after the use. So this is also just handling with care. Using the light microscope. How to use the light microscope? It's just these are just procedure of how to use the light microscope, and then we calculate some calculation here. Number one, you just place the microscope on the bench with the stage facing with the from you. Then turn the lower power objective lens until it clicks on the position, meaning that it produces a sound. So it produces a sound. So it produces that sound, meaning that this thing it is in position. Ensure that the diagram is fully open, because the diaphragm is fully open because the reason is to allow enough light to pass. Look through the eyepiece with one eye. Meanwhile, adjust the mirror. You have to adjust the mirror. So place the slide containing the specimen on the stage. Again, look at it. That's a simple procedure, and this is not that common question. It's not that common question. So study question. How is the low objective lens manipulated to focus a specimen for observation under a light microscope? How is the low objective lens? Because this question is from here. It's from here. It's from here. And the low objective lens until it clicks into position. So how you make sure that it clicks. So click the lower power objective lens in the position. So to make sure that it clicks the lower power objective lens in position. So you use the rough cost adjustment knob. Cost adjustment knob. So with eyepiece in the two eyepiece lens and using the cost adjustment knob, gradually raise or lower the objective lens, bringing the specimen into focus. So the answer up to here it is very correct. This is just explanation. Give a reason why only fine adjustment knob should be used when using high power objective lens. High power, or give a reason. This question is tested in two ways. Give a reason why only the fine adjustment knob should be used when using high power objective lens? That's the question one. Question two is give a reason why cause adjustment knob should be used when using should not be used. I mean, should not be used to lower the high power objective lens. They are two different questions, but the answers are the same. Answers are the same because uh, the high adjustment knob, uh, the fine adjustment knob should be used when using high power objective lens, or coarse adjustment knob should not be used. Reason is to avoid breaking the slide. That's number one. Number two is to avoid destroying the specimen. Difference between the light and the electron microscope. Light microscope uses a light to eliminate the specimen. This one uses the beam of electron. The light microscope uses glass lens, uses magnetic lens, electron microscope. It has low resolving power. Resolving power is just the resolution, the ability to distinguish two structures that are very close together as distinct entities. Meaning that this light microscope it cannot distinguish the it does not have that much ability to distinguish. But this one has very high ability to distinguish it. This has low magnification. This very high magnification power. This has the specimen under view may be alive or dead because the reason is this is using the light, natural light. So the natural light will not kill the specimen. So the specimen might be live or dead. But this in under electron microscope, the specimen viewed and under view is must be dead and is dead understand because it's usually dead because it's using beam of electrons that kills specimen that kills the specimen. So these are questions, some part of the microscope. This is the eyepiece lens, rough adjustment knob, smooth adjustment knob. This is the arm. This is the objective lens. This is the mirror. This is a stand. So that's all about it. Modification of the microscope. So when we're talking about modification, is the ability to enlarge, ability to enlarge, uh, ability to enlarge object so that it can be seen through the naked eyes. So in microscope, we have two lenses. That is the eyepiece lens where you place your eyes, and objective lens where you place just above there where you place the specimen. So magnification is calculated by using these formulas. Magnification is equals to eyepiece lens times objective lens. Eyepiece lens times objective lens. Look at this. For example, this question, total magnification will be 10. That's the eyepiece times objective lens, which is 5. So 10 times 5 it is 50. This is 50. This 5 times 4 it will be 20. This one we were given total magnification 600, and this one will be given we were given objective lens. Eyepiece lens, how can we calculate? We calculate by using this. We say that magnification is equals to eyepiece lens times objective lens. So now we have the magnification which is 600, which is 600 is equals to the eyepiece which we don't have times objectives which is 40, which is 40. So this will be 40i, 40i. So divided by 40, divided by 40, divided by 40, divided by 40. So this one will be 15, x15. And then when you are writing modification, you must write the x before the number because how you can know this is modification. Because when you write only 15, 15 is just a number. So you must give the units, and unit is x15. So that's all about the microscope. That's all about the microscope.